main macromolecules in our cell, which we already came up with: DNA, RNA, proteins, and glycans. Almost always are copolymers. Which one is the only one that would probably not be a copolymer? Glycans. You could have the same sugar the whole way down. I'm gonna go ahead and venture to guess, and I can't say this definitively, but there are no proteins that are like lysine the entire way. There are no DNA strands that are G, the only, and that's it. Like, I mean, so I'm sure this exists somewhere. But what we're gonna talk about is almost always gonna be a copolymer, right? So with DNA and RNA, we can have 4 types of monomers. And again, I've characterized them by their base, but the nucleotide also has other parts to it. It's not just a base, it's a base attached to phosphate groups and a base attached to a ring of carbons. We'll show that structure later. But we're mixing potentially 4 things with each of these: adenine, thymine, cytosine, guanine, or uracil. Proteins have 20 types of monomers. Yep.
I was just wondering, with the lipid bilayer, like since it's made out of multiple fatty acids, would that not be a polymer?
It is not, and somewhere in one of these slides it says lipids, not a polymer, literally. So, exactly right, perfect. So proteins have 20 types of monomers. There are amino acids. Glycines have lots of possible units. There's a lot of different types: fructose, sucrose, whatever. There's variants on those sugars. It's really not the focus of this class. Don't worry about memorizing those. I think that discussion makes up the first few sections of chapter 3, and on the syllabus it says optional reading. I'm not going to go into it in detail, so unless you're really into glycans, I would maybe focus elsewhere. The types of, from chapter 3, what we'll focus on is the lipids, and lipids just don't really happen to fall into the polymer category. Okay, so copolymers are seemingly random for our biological molecules. But what is a gene? First of all, which of the macromolecules does gene belong to? DNA. Okay. And then who wants to give a definition to it? That's all right. I'll give it to you. It's a segment of DNA that codes for either functional RNA or messenger RNA, and then the messenger RNA would of course be coded for the production of a functional protein. Okay? So while copolymers are seemingly random, and usually that sequence of bases is going to look random to us, remember that genes have a specific sequence that's very meaningful to what it codes for. Okay, so it might be a random mix of A, T, C, and G, but it's really not, because when one of those things gets screwed up, it leads to downstream problems in the synthesis of RNA or protein and protein culture. Okay, so there's this word functional. The textbook talks about functional RNA and functional proteins. Is DNA functional? No. So what's a word we might use instead? Or we'll use the word the book uses. It's informational. They say it's carrying information. RNA could be either. RNA could also just carry information or instruction. DNA is always carrying that information and instruction, and proteins are always going to be the functional kind of final product. I think I put, oh, this is a comment I didn't bring up yet, but we can stop and chat about it if people are still copying notes. I will make all of my slides available always online. I almost always will make them available after class. And the reason is, I like edit on the fly while I'm presenting them. I may skip stuff and be like, nope. And I don't want to waste my time putting it up there and having people read it later when I've decided we're not gonna talk about it. So what I can do after class is edit them, and then they'll go up looking, especially if you catch errors, which you will. I can fix them before I even post them, which is nice. The other thing that I will say is just a huge hint for everyone who's going to go through and look at my slides later. The notes section usually has the answer to any question that appears on the slide. So if you weren't here and you don't watch the video, read the notes part when you're in PowerPoint. Don't, display it as a slideshow. Read them, and then there's also extra information. So for example, this one has for me an example of a functional protein is hemoglobin. What is its function? Transporting oxygen. It's going to bind oxygen in the lungs, circulate in the blood through to peripheral tissue, and drop off the oxygen there, right? Easy one that most of us has heard of, has a function, okay? It's a protein. So like I said, that little blip of description is, I will leave it in the notes, and those are a good place to look for stuff. Stuff I might ask about in the future. Okay, interactions between these four main macromolecules are the basis of all biological function. So, how proteins interact with each other, how proteins come back and bind onto DNA and do something to it, all of that. Proteins and glycans can come together, can graft together into a glycoprotein, and then that's going to have its own unique function. So again, some type of interaction between all of these things drives all of the function in our body. So we can have interactions in the polymer backbone itself. What types of bonds are in the polymer backbone? Covalent. Beautiful. That is the word I'm looking for. What about interactions between molecules? So between two polymer chains, or between my DNA chain and my other DNA chain, my DNA chain and my protein. Hydrogen, van der Waals, all types of
Secondary bonds.
secondary is a great word. Non-covalent is the word I was going for, and all those were examples of them. Yes, these are some type of non-covalent secondary interactions. They could be attractive, they could be repulsive, they could be stable, they could be transient, so they maybe come and go. There's binding and then it kind of comes apart quickly. They are always weaker than covalent bonds. Okay, so the backbone is much stronger to kind of break apart in an interaction between two molecules. We want to understand the thermodynamics and the energetics of these secondary non-covalent interactions for many reasons, but the two we'll talk about: a better understanding of the biology of the human body, particularly to treat pathological conditions. Okay, that's our goal, is to help people in the long run. Okay, so protein misfolding kind of underlies a lot of pathological conditions. Okay, and a misfolded protein could result from some problem way back in that DNA genetic sequence, right? One of the bases was wrong, it led to a different RNA transcription, it leads to the translation of the wrong amino acid, and that's why it can't fold. Or it could have something simply to do with other things related to these secondary interactions. Okay? So it's not able to make some type of hydrogen bond or van der Waals bond or something that it needs. And the other reason would be biomimetics. Okay? Biomimetics is innovation inspired by nature. We want to understand nature, we want to understand biology, and basically engineer something for ourselves that utilizes those principles. And we'll do some examples in class. But I threw in my notes one that I don't bring up later, so we can talk about it now. Has anyone ever heard of like a thermophile or an extremophile? What is that?
Organisms that can live in extreme conditions.
Exactly. An organism, so extremophile is kind of a broad term, right? It's an organism that can live in some type of extreme condition. Thermophile means it's living at extreme temperature, either really, really hot, like those thermal vents in the ocean, or possibly somewhere very, very cold. It lives in the ice in Antarctica. Other types are like acidophiles, bacillophiles. They can live in extreme pH conditions. There's a whole bunch. So we want to study these like organisms. We want to study exactly what types of proteins they have and what types of interactions in the proteins are so stable that they can still function in these conditions. Can we recreate and like protein engineer something for ourselves that can survive something similar? Okay. And I think that's a cool topic, so I may bring it back up later when we talk about proteins with some more specifics.